my talk, we're going to get to the very, very details of the molecule. I hope that I don't put everybody to sleep, but it's going to be short. So we're going to talk about in general, about the structure of KIF1A and then of the particular mutant P305L. So this is the beginning of this work. And I would like first, before I have time, is to acknowledge the people who have been working in this project. This is a collaboration between the lab, the Army Generate, and my lab. And the people doing the work has been Lou Real, Matthew Benoit, and Ana Asenjo, who is also here today. So, as it has been mentioned before, kinesin is a big superfamily of motor proteins. And the common characteristic of all the family is the engine, the little thing called the motor domain, which has a structure as shown here on the left side. And this motor works on a particular track on the cell that is called the microtubule, which are a tubule that goes along the cell that are part of the cytoskeleton. And the superfamily has like 14 different families. We have been in my lab studying many members of the family. So KIF1A is in particular belong to the kinesin 3 family. So this is a scheme that is valid for all kinesis that are motile, that are most members of the kinesin superfamily. They work in what is called a hand-over-hand mechanism, in which the 2 feet or the 2 motor domains alternate as they walk along the microtubule. And the energy that is necessary to provide so that this molecule can do this work is provided by ATP, which is called the coin, the energetic coin of the cell. So each feet hydrolyze ATP, and that they give the energy to produce movement. So what I have said is general for all the kinesin, but KIF1A has a few characteristics, like they have some part of the structure that are unique to KIF1A. One of them is called the K loop, and we're going to talk more about that's important in the structure. So, as it has been mentioned, there are many, many mutations. That means we have in the motor domain about 300 amino acids or 300 letters, and a change of each of one of these letters produce one of the CAN-associated diseases. And there are many, many in the motor domain. And we're going to talk in particular of one, P305L. So what happened with this mutation? Well, you can imagine this motor work like somebody moving a monkey bar. So in order to do that properly, you have to have grip, have to have force, you have to have certain velocity to get to your destination at the right time, and you have coordination, because if you let it go both hands at the same time, you fall off from your track and you never get to your destination. So coordination is also important for this motor to work. So the approach that we are using is we are determining the structure of the motor and of the mutant to try to understand what is going on. And the approach is analogous to what happened when you have the broken car. The first thing that you will do is to open the hood and see what is wrong, to try to find a way that maybe you can fix the problem. So here is the structure of KIF1A. So this is the crystal structure of KIF1A obtained many years ago, but this represents the protein. In this view it's not bound to the microtubule. And when I say the structure, there is not a single structure, because these are molecular machines that they change the structure as when it binds to the microtubule, when hydrolyze ATP, and when it moves, there is an arm that is going to move to the next position. So all these conformation, all these changes are different structures. And the idea that we would like to do is to understand what are all these structural changes in the normal protein and how they are changed in the mutant protein. So in the crystal structure, what we have is a view of the molecule when it's free, not bound to the track, to the microtubule. And some areas of the structure were not visible because they were very disordered or mobile. And some of these areas are regions in where some CAN mutations are located. Here I just pointed out in red P305L. So the rest of the area, which is they call the K loop, is not visible in this crystal structure. And we would like to know what happened with this area. So we use cryo-electron microscopy to solve the structure of these proteins bound to the microtubule track. So cryo-EM is a microscopy, use an electron microscope, and it's a technique that in the recent year has done tremendous progress, and now is able to give almost atomic resolution. The atoms of the whole protein resolve when it's bound to the track and sometimes when it's doing work. So we resolve the structures of KIF1A, the wild type first to have a baseline in different states of the nucleotides. Like if you were an engine and you freeze the motor at different times of the piston moving up and down. So for doing that, we use analogs of the molecule of energy of ATP and use different analogs to produce different structures. So we remove the analog that is called APO, we use ADP, or we use AMPPMP. And we obtain different structures in each case. We use the dimer molecule, and when we use ADP or APO, we get only one of the heads bound. So you can imagine this is a state when when one of the feet is bound to the microtubule and the other is free to move to find the other position. While in the other case is the case when both heads are bound to the microtubule. Interestingly, we found that the structure, though, is not easy to see in this picture, but it was different between the two heads, which that indicate that they are coordinated because they are in different points of the ATPase cycle, and that's an insurance so that the molecule can never gonna release both at the same time, or if it's gonna do it, it's gonna do it with very low probability. So another way to look at the structure is in these diagrams called ribbon diagram. And here we are looking at the molecule from a side, and the microtubule on the left side. One thing that we just found out is a new structure for this KIF1A. This open, what I call an open structure, we found a structure in the nucleotide is complete, nucleotide binding pocket is open, and that explains one particular point of the ATPase cycle of this protein. So that only occurs on the microtubule, and that's why it wasn't being observed before for this kinesin. So, and to summarize, and to bring, here you see the complete cycle of conformation of one motor domain. So starting from the free kinesin, it binds to the microtubule, something move that open this pocket, ATP, which is the orange molecule, it gets released, and then ATP comes back. And then when you see this little R moving in the back, this is actually what it moves the other head forward. So that's the forward movement that happens when the ATP molecules bind. So these are the cycle of conformational changes that each head goes through as it walks along the microtubule. So that all this structure gives like the base to try to understand now what happens when you have mutation. What does it change? And we started with I forgot. We also resolve. I mentioned before one of the characteristics of this kinesin is that they have this called the K loop. You see on the left side this pink round thing that goes, that's unique of KIF1A, and it's a very positively charged area that helps this motor to grab to the microtubule. So that's give this particular kinesin a particular is, let's say, a solid grip into the microtubule. And mutations in this area affect how the motor actually binds to the microtubule. This area, it wasn't clear what is binding, and it's interesting that here we see the two kinesin in blue on the side, and then maybe. So here this is the structure of the two motor domains connected by these red lines called the neck linker. And this is the K loop. This is the unique for kinesin 13. And what we see is that it doesn't interact with the core of the tract, the microtubule, but with this kind of floppy area that are colored here in yellow. And if we color by chart here, these are called the C-terminal tails of tubulin. They are very possibly charged and very flexible. And they interact with the K loop, and that provides kind of a mobile grip that allows the kinesin to bind, to find the place on the microtubule and move to the next one without actually leaving the vicinity of the microtubule. So another interesting thing that we found, we have worked with other kinesin, but different KIF1A has a very tight connection between the two motor domains. This is another kinesin, and you can see that this connection is kind of very loose. And we have looked into other kinesin which this connection between the two heads is longer. So we believe that this very tight interaction between the two heads help the two motor domains to be coordinated. Coordinated means one motor kind of talking to another to tell them, don't let loose. I need to move to the next position. And that coordination helps this motor to move for very long distance, which is a characteristic of this kinesin, KIF1A. So we did the same with P305L, and we basically see the same conditions to the wild type. Like in this condition, we see only one head bound. In AMPPNP, we see the two head bound. However, as has been reported by other people, the binding is very, very weak. We have to put a lot of kinesin close to the microtubule in order to even see it bound to the microtubule. So the affinity of this protein for the microtubule is a lot lower. That only for, and this is caused just by this mutation P305L. If we look in detail around the mutation site, this is a view like if you were the microtubule and we're looking at the kinesin. In other words, if you will be putting yourself behind your car to see what's going on the bottom of your car. You're seeing the interface. And this is where the P305, the proline 305 is located in the wild type. So this is the structure of the wild type, and this is the structure of the mutant. And one thing that it was the conformational chain or the structural chain is relatively small, a lot small than we had predicted. Based on other studies, people have thought that this area here that is also part of the microtubule binding site will change, because this is an area that is conserved in all kinesin, so it's supposed to be very important. But we see very little change here. And then we see that these residues on this side of the molecule, the N-terminal, it does change position. You see phenyl F303 or phenylalanine 303 moves from this position to this position. And here we have an overlap to see the difference. So it is a small difference, but it's caused by the introduction of this leucine residue that makes a clash with this bulky aromatic ring, so that move it away. So looking at the structure, it made you think, well, if there could be a way that we could fix this problem. And as a proof of concept, we thought, well, maybe if this put a less bulky residue here, we could perhaps correct the problem. And we did that. We mutated this residue to a valine, which it doesn't have this bulky aromatic group here, and see this functional assay to see what happened. And it does revert a little bit. So this is the wild type. Here we are measuring something called the run length, how long this motor can walk. We just measure a lot of motors walking and see how long they go. And this is the wild type. This is the P305L mutant, and you see a huge impairment. It can go for very short distance. When we have the double mutant, we get a little bit of improvement. So that means that perhaps altering the environment where the mutation is, it could restore part of the function. It is not complete because the mutation that we use, the 303 by itself, cause a problem. So this is by no means an ideal solution, and also it's another mutation, but it's just to show that perhaps a molecule, a small molecule that binds to this area, could revert the effect of this mutation, and that would be possible. So this is a view, exactly the same view of the area where the mutation is. This is the wild type, and this is the P305L. It's colored by charge. And what you see here, it is the microtubule, the track, and you see here is the kinesin. And you see, like, it is a very tight interaction. You see very, very nice complementary here, and you see like a pocket here. When with a mutation, you see that the interface is altered. You don't see that nice complementarity, and that do explain why the binding of this protein is not that good. It also shows a pocket that perhaps could be exploited to bind a molecule that it binds specifically here, and perhaps helps this protein to bind to each other. So I'm about to finish here, but as I said, there are many mutations on the motor domain, and this is what we only find one here that is in this location. Our goal is to find the structure of other mutant to see what's wrong with the structure. It seems like a daunting task when you see that many places that it could be mutated. However, when you look at the structure, you can divide the motor domain in areas. For example, this is the interaction, the microtubule interacting region. So all these regions, you could say, are going to affect microtubule binding. Here is the nucleotide binding, so that may affect how this molecule hydrolyzes ATP. There are other regions that are not close to any particular functional area, but we know from our water kinesin that this machine works by moving different domain. So it is possible that some of these mutations, what affect is the movement that this molecule have to do to proceed and to do work. So the hope is that we could buy classes of mutants. So we want to start from a few mutants here, a few mutants there, and a few mutants there, and find like motifs that perhaps could cover or drugs that could cover a range of mutations in the same area. And we just happy to say we just got funded to actually do this work. NIH grant just got approved to start doing a structural work on these other CAM mutants, and we are happy to say that. And that's about it. I can read the conclusion, but so we found a novel open conformation, as I say, for the wild type kinesin. We saw this K-loop that is specific for KIF1A. We also saw that it had a very tight connection between the two motor domains that may explain part of why this motor can work for such a long distance. And we see what are the structural consequences of have this P305L mutation on the structure of KIF1A. And so with that, I'll just put again the acknowledgement and happy to.
Going back to the other sentiment and with George, that he was saying that KIF1A may be implicated in Alzheimer's because it interacts with that C terminal in the tubulin, that you could just resolve the interacts with the K loop.
Yeah.
I don't know, that would be, I'm loud enough.
That would be an interesting idea to explore.
Yeah, I mean, certainly the idea will be as use that for in silico drugs screening or actual drug screening, and then to improve. If you find a drug that binds to a particular pocket, it will be to, you know. Yeah, well, I mean, in general, finding drugs that will affect kinesin. I mean, this approach will be, that's what we would like, something that it could affect the grip of the kinesin, the velocity of the kinesin, and maybe even if it's not specific for KIF1A, it could be helpful because, you know, making them a model to bind better, that could help KIF1A and perhaps other diseases too, associated with other kinesins.